वेलकम बैक टू आवर चैनल बी क्यूरियस। आज हम पढ़ने जा रहे हैं वाई जीन क्लोनिंग एंड डीएनए एनालिसिस आर इम्पोर्टेंट। इन द मिडल ऑफ द 19th century, Georg Mendel formulated a set of rules to explain the inheritance of biological characteristics. The basic assumption of these rules is that each heritable property of an organism is controlled by a factor called a gene. The early development of genetics. For the first time, 30 years of its life, this new science grew at an astonishing rate. The idea that genes reside in chromosomes was proposed by W. Sutton in 1903 and received experimental backing from T. H. Morgan in 1910. Morgan and his colleague then developed the technique for gene mapping, and by 1922 had produced a comprehensive analysis of the relative positions of over 2000 genes on the 4 chromosomes of the fruit fly Drosophila. The advent of gene cloning and the polymerase chain reaction. The methods refers to as recombinant DNA technology and genetic engineering. The whole new method biology was developed is enabling previously impossible experiment to be planned and carried out, if not with ease, then at least with success. They led to rapid and efficient DNA sequencing techniques that enabled the structures of individual genes to be determined, reaching a culmination at the turn of the century with the massive genome sequencing projects, including the Human Project, which was completed in 2000. Construction of a recombinant DNA molecule. Vector fragment of DNA combined to form recombinant DNA molecule. This basic steps in gene cloning. Construction of recombinant DNA, transport into the host cell, multiplication of recombinant DNA molecule, division of host cell, numerous cell division resulting in a clone. What is gene cloning? What exactly is gene cloning? The easiest way to answer this question is to follow through the steps in gene cloning. A fragment of DNA containing the gene to be cloned is inserted into a circular DNA molecule called a vector to produce a recombinant DNA molecule. The vector transports the gene into a host cell, which is usually a bacterium, although other type of living cells can be used. Within the host cells, the vector multiplies, producing numerous identical copies not only of itself but also of the gene that it carries. When the host cell divides, copies of the recombinant DNA molecule are passed to the progeny and further vector replication take place. After a large number of cell division, a clone of identical host cell is produced. Each cell is the clone contains one or more copies of the recombinant DNA molecule. The gene carried by the recombinant molecule is now said to be cloned. What is PCR? The polymerase chain reaction is very important, very different from gene cloning. The basic steps in PCR experiment are follow: denaturation of the template DNA at 94 degrees Celsius, annealing of the oligonucleotide primer at 50 to 60 degrees Celsius, synthesis of new DNA at 74 degrees Celsius. Repeat the cycle 25 to 30 times. The mixture is heated at 94 degrees Celsius, at which temperature the hydrogen bond that hold together the two strands of the double-stranded DNA molecule are broken, causing the molecule to denature. The mixture is cooled down to 50 to 60 degrees Celsius. The two strands of each molecule could join back together at this temperature, but most do not, because the mixture contain a large excess of short DNA molecules called oligonucleotides or primers with anneal to the DNA molecule at specific positions. The temperature is raised to 74 degrees Celsius. This is a good working temperature for the tag DNA polymerase that is present in the mixture. The temperature is increased back to 94 degrees Celsius. The double-stranded DNA molecule, each of which consists of one strand of origin molecule and one new strand of DNA denature in single strands. This begin a second cycle of denaturation annealing synthesis by repeating the cycle 30 times. The double-stranded molecule that we began with is converted into over 130 million new double-stranded molecules, each one a copy of the region of the starting molecule delineated by the annealing site of the two primers. Why gene cloning and PCR are so important? Because both technique can provide a pure sample of an individual gene separated from all the other genes in the cell. The mixture could indeed be the entire genetic complement of an organism. A human, for instance, each of these fragments become inserted into a different vector molecule to produce a family of recombinant DNA molecules, one of which carries the gene of interest. Cloning follows. Cloning allows individual fragments of DNA to be purified. Vector plus DNA fragment construct a recombinant DNA molecule. Each carries a different fragment. Introduced into bacteria, played out. Each clone contains multiple copies of just one recombinant DNA molecule. PCR can also be used to purify a gene. The polymerase chain reaction can also be used to obtain a pure sample of gene. This is because the region of the starting DNA molecule that is copied during PCR is the segment whose boundaries are marked by the annealing positions of the two oligonucleotide primers. If the primers anneal either side of the gene interest, many copies of that gene will be synthesized. Because the desired gene is automatically selected as a result of the positions at which the primer anneal. PCR has two limitations. In order for the primers to anneal to the correct positions either side of the gene of interest, the sequence of these annealing sites must be known. It is easy to synthesize a primer with a predetermined sequence, but if the the sequences of the annealing site are unknown, then the appropriate primers cannot be made. There is a limit to the length of DNA sequence that can be copied by PCR. 5 kilo base can be copied fairly easily, and segments up to 40 kb can be dealt with by using specialized techniques. But this is shorter than the length of many genes.